Okay, today we're going to start a part where I am showing you the different types of alignment. You are familiar already with BLAST2. The L in BLAST2 is local, but there are other ways that we could align different sequences. And the motivations to use local, which is what you used, versus global, you're going to learn those in class. And how do you calculate the score from one alignment versus another? They use different algorithms, so different sets of instructions, which were developed by some mathematician or bioinformatician, whoever it was, taking the 80s, 70s and 80s. And you're going to see their names because the algorithms are named after the people that developed them. So let's see. I want, no, it's file. That's what I want. Share files. This one. All right, so you already learned one type of sequence alignment, which was BLAST. We used BLAST2 so far, but there will be BLAST in the future without the 2. We can compare sequences pairwise, meaning you are just comparing two things, one read versus another. Or you can do something called multiple sequence alignment, which is I have this group of 3 or more sequences, and I want to know what they have in common. In its core, this multiple sequence alignment starts as a pairwise, because when you're comparing 3 things, you are really starting like, okay, I have these 3 strings. I'm going to look at the first letter of one sequence, and then the first letter of the other sequence. So really it starts as a pairwise, as you will see. But the algorithms we use, like the software, the programs we use, they are different for pairwise versus multiple sequence alignment. And a lot of it is because the goals are different. When you do pairwise, maybe all you care about is to see, do they have any chunk of sequence that it looks alike, that is similar? Which, for you, I showed you it's when we are aligning two reads and building a config. So we know that some part of it doesn't overlap, and then another part of it overlaps. And we're just trying to find the overlap to put them together. And so that when we do the pairwise, we are looking, are there blocks? So you're going to see that pairwise normally does local alignments. Are there blocks of similarity? And when we do multiple sequence, we're just seeing overall, how does one sequence resemble the other? So the multiple sequence alignment normally uses global alignments. Right now, you don't really know the difference, but we'll do an exercise comparing these. And you want to have this written down somewhere, because in homework and then in exam, I ask you, what's the name of the algorithm that does local alignment? And it's these two guys are the ones that developed that. And what's the name of the algorithm that does global? And it's these two guys here. I don't actually know how to say this name, but I'm going to say it the way it sounds in my head. It might be wrong. Okay, so what do I want you to learn from the lecture? I want you to know that there are different kinds of alignments. So either you're comparing two sequences, or you're comparing multiple sequences, so 3 or more. Maybe you're comparing the different strains of COVID that exist, and then you want to see where they're the same and where do they differ. Then you'd have 3 or more strains that you're comparing. Then there is local, which is you are zooming in some spot and seeing is this part, do they resemble each other? And then the global is more like overall, what do they have in common? So what are we learning when we do alignments? And how do you know if you're going to choose local or global? But it's mostly here looking, if you're looking for so some parts of overlap, you do local. If you're wanting to compare overall how they compare, you do global. And we might get to this today, we may not. So there are these words here that if you stay in this field of bioinformatics or molecular biology, you're going to hear homologs. I remember when I was a graduate student, um, many seminars. I don't know, alignment was so in vogue. Everybody, because I guess the tools had just come out and everybody was doing alignments, and they were proud to show we have these sequences and we found a homolog of this gene. And I remember initially going like, what is that? So you guys are going to know. So what is a homolog? And then this ortholog and paralog are types of homolog. So you're going to learn when it's this one and when it's this one. And then this is really toward the end where we're going to be doing an activity you're probably not going to finish, and you're going to pick it up on Monday. So, yeah. So the sequence alignment, we are lining up DNA against DNA sequence, RNA against RNA sequence, or maybe if you have DNA sequence for one organism and then RNA for the other, the program is probably not going to complain. It knows to make the necessary changes. But you cannot compare RNA to protein. That does not make sense because protein sequences are made up of amino acids, and there are 20 kinds of amino acids. DNA sequences are made up of nucleotides. There are 4 types of nucleotides. RNA sequence is made out of 4 types of nucleotides. So if you compare an RNA sequence to a protein sequence, the computer would think or would assume that you only have those 4 nucleotides or those 4 amino acids. It would think everything is protein, but it would probably flag it. They write this program that if the computer sees only 4 letters in one sequence, it will say you probably didn't enter an amino acid sequence here. So when we are doing alignments, we compare protein against protein, and then DNA against DNA, RNA against RNA, or even these two against each other. And here are the types of alignments, and maybe I'll show you now an exercise of let's think what is different. So the local is looking for blocks of resemblance. So if I am doing pairwise comparison. So I'm comparing this lady and this dog, and then this lady and this dog. If I am doing this locally, well, actually, I already gave you the example. If I'm doing this locally, maybe I'm focusing on the hair. So that hair with the hair of the dog. So if I am trying to build a phylogenetic tree, so the phylogenetic tree is trying to say these things are closely related. So if I am looking at this and I'm making my phylogenetic tree, and I want to make it like, okay, I want to group my species together, then this lady and this lady would be in one clade, so in one branch of the tree. And then this dog and this dog would be in another branch, because we're gonna go people in a branch, on a clade, and then animals on the other clade. So when I'm doing phylogenetic trees, I'm looking like overall, I'm not zooming in. But if I'm doing a local comparison, I might be looking at, like I said, just the hair, or maybe like the body color. So I'm just looking at zooming in some spot, right? So there are reasons we do that with sequence. And as you guys have done in the past, maybe I just want to see, do they have a block of similarity? I don't want to see how they overall resemble each other. So did you get like the main idea of this? All right, so good, thank you. So when you're comparing the hair color, this would be, you guys tell me, local or global, if I'm zooming in on the hair color. Right, Sydney? So yeah, local, good, Michael, local. And if I am looking at the overall resemblance, then that's global. Good job. So here, let me think. Yeah, well, normally I would stop here and make you do this exercise. but I think this will be harder to do from in our current environment. So I will show it to you. Normally I want you to suffer through it, but anyway. Okay, this Needleman, okay, I don't know, Wunsch, whatever. So these guys, they developed the algorithm to do global alignments. And these guys here, Smith and Waterman, I think this was first and then this is second. It was an adaptation, so a change on the rules that these guys created. So these guys tweaked something and they're like, oh, look how magically it does something different. So here's an example of sequence. The global one, and you're going to learn how the scoring is built, which is kind of exciting to know that there isn't magic the computer does. It's just following your directions. And then it comes up with this alignment here. What it's doing is F matches F. Great, keep them aligned to each other. It could have matched this T over here, and sometimes it does make different alignments with the same two sequences, even if you do both global. So this T could have been lined up with this T or with this T. And in this case, the algorithm preferred to put a gap here and shift this T that was here to match this one here. And okay, then the F doesn't match anything, so it was best to put a gap there. You're going to do something called dynamic programming. Ah, yes, thank you. So Helen said, basically, she's like, how about you use your little mouse? Thank you, Helen. When I'm sharing my whole screen, I'm assuming you see my mouse. But thank you. Yeah, so this F matches this F. And then the next thing that the computer is going to look is, does this T match the next letter of this? And it does match this T here. So it could have been shifted to here and then a gap here and here, or the computer calculated a higher score or a neutral score by shifting the T over. So it says that matches that, then gap, gap, match, 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 put a gap here, shift the L that way. So the global is showing to you how they overall resemble each other. What global alignments try to do is to stretch the analogy or the alignment between the first and the last, in this case, amino acids in a sequence. And then for local, it's trying not to stretch. It's doing the opposite. It's trying to keep the letters as close together as possible, even if the beginning or the end of the sequences don't match. All right, yes, see that? And then you might wonder, why would I choose one over the other? It depends on your goal. If you're doing phylogenetics, you're using global. If you're just looking for, I have this really long sequence and then this other sequence that maybe if it's a short sequence, you know that overall they're not going to resemble each other. Just looking to see, do they have chunks that are in common? So far so good. So here's when I would not show you the answer, but I will, because this actually takes time, and normally I'm just walking around and making sure you are awake. But I would make you align this sequence and this sequence. And that exercise is in that Word document. But here is already the answer, which if you're looking at this, I tried to give you some hints, as in I made this like italicized to show you that that matches here. And in the end, you would figure out, okay, if this part italicized matches this part italicized, and I keep those together, I'm doing a local alignment where I'm seeing what chunk do they have in common. And in the global alignment, I'm just trying to stretch and sticking the gaps wherever I need to see how they overall, from the beginning to the end, they resemble each other. So here are the whys we do them. You did this one before. We assemble the sequences in contigs. you're doing a local alignment here because we're doing something, let me go back, something more like this. This would be very annoying for assembling that this chunk didn't match this, but for the exercises that I gave you, they align perfectly, and it's, I make it so that it works. In real life, you could have things that don't align when you're assembly config, and it could be that this chunk of DNA is a repeat, and therefore this exists in some chromosome. It is on exists in another chromosome, and you shouldn't actually make this into a config. But in our exercises, I make it so that they line up perfectly. And you might be happy to know that I'm not going to ask you to make any more contigs. So that is in the past. If you're like, okay, I didn't get it, don't worry about it. If you work in this field, you're going to have software that you throw all the reads in there, and it does that job for you. All right, the other thing that we use alignments for is to identify a sample. When I worked in a company, we would sometimes, okay, let me just stop sharing this for a moment. So I worked in a company when I finished my PhD that we received samples from all over the world. And it was a flower company. And sometimes there were some viruses that were infecting a crop, and we didn't know which virus it was. So my job was to extract often RNA, because lots of viruses are RNA virus. And then I would clone, or I would revert that to DNA, take that DNA fragment, put it in bacteria, send that off to sequence, and then I would receive a text file back, and I would say, okay, I have this sequence that is something infecting the plants, and I don't know what that something is. Maybe it's the first time this ever occurs. And that happened so often in the company, the first report of this virus in this plant. And then I'm like, my gosh, we could publish a paper every other week with this information, or at least like a note. But we were not allowed. There was like a secrecy thing. So yeah. So then I would get the nucleotide sequence, and now I want to know what is this nucleotide sequence. So I would use an alignment to say, this is the sequence from the plants, and then compare that to a large database of all the nucleotide sequences that we know where they came from. And then that's how I would identify what was infecting the plant. So that type of alignment, and you guys are going to do that in class, that's BLAST. Remember in BLAST 2, you give both sequences and you see what do they have in common? Well, when it's just BLAST, you enter one sequence, and there is a large database that BLAST is going to compare your one sequence that you entered against everything else. So this is a really good skill to have, and it's good for you to know. I don't know, if you're going to have an interview, that you know what BLAST is. I don't know, for me it's like one of the coolest and easiest tool to apply for like real life. All right, so share that.
Mom, can I eat at 11:45? Okay, thank you.
All right, let's see. So the reasons why we do alignments, here we are. All right, so the identifying samples, it's you have some nucleotide sequence, and maybe you want to know where it came from. We do alignments to do phylogenetic trees. This is some example of, we're basically looking how alike, if you're going to group things, we want to group which ones are more alike to the others. So in this case here, you are doing a multiple sequence alignment. The identifying samples, you are doing the one sample against one sample in the database. And then is it a match? Yes, no. Then your sample, another sample in the database. Is it a match? Yes, no. So this one would be a pairwise. And other reasons why people do alignments, they want to find homologs, which here's the definition. Homologs are sequences that have a common ancestor. So if they have a common ancestor, they may still be the same sequence, or they may have some mutation. And there are different types of homologs. So orthologs, you are comparing, okay, this homolog in one species against a homolog in another species. And those two species have some ancestor in common, and that's why both of them have that sequence. So when you're looking at orthologs, you are looking at, does that sequence exist in different species? Paralogs are sequences that underwent some duplication. So you can think like we have paralogs. Let's see, the most common example is globin. We have three versions of globin. So there were duplication events, and so there's a duplication, and then there is some changes. That's why they're not exactly the same. But you could think about this as paralogs is a sequence that appears in parallel within the same genome. So in that organism, there is a duplication of that sequence, which may have undergone so many mutations that they do similar but not exactly the same function. Okay, so other reason we find consensus sequence. So consensus, you could think about that as you're ordering pizza and everybody tells what's their favorite topping, and you find the consensus, and that's the sequence or that's the pizza you order. In terms of sequences, you're trying to see which nucleotides or which amino acids were preserved in all the different things that you're comparing. And lastly, we use alignments to predict gene function. So you want to see if I have this DNA sequence in one organism, does it do the same thing in another organism? Because maybe I know what it does in one organism, and I could speculate it does the same on another one, but it doesn't necessarily. So the predicting gene function is not, it's not proving that the gene function. You don't actually know. But from the bioinformatics, you can say, since it does this in one organism, it might do the same thing in this other one. But the only way to actually find out is to make mutants in the lab, but it's much more laborious than just predicting, sitting in front of the computer. So actually, let me pause here and see if you can do an exercise with you guys to get you doing something.